is Mark Hildebrand. I'm a research scientist and the director of the Marine Biology Research Division at Scripps Institution of Oceanography. I'm going to talk today about the diversity of algae and specifically about a class of algae known as diatoms and brown algae. So in the first slide here, we're seeing images of cultivation of two different classes of algae in a photobioreactor. So on the left, we have green algal culture. Green algae are also known as chlorophytes. And on the right, we have a culture of diatoms, which are known also as stromatophiles. And in these photobioreactors, we're illuminating them from the top to mimic sunlight. And you can see a very drastic difference in the pigmentation of these two different classes of algae. Green algae obviously are very green. The diatoms are golden brown color. And the reason is that these algae are very different from each other, and it stems from their evolutionary history. So as near as we can estimate, about 1.5 billion years ago, a unicellular eukaryotic heterotrophic organism that could not fix carbon engulfed the cyanobacteria. So the cyanobacteria were the first classes of algae that could fix carbon through photosynthesis. So during this engulfment event, the cyanobacteria eventually evolved into what we call now the chloroplast, and we had the formation of an initial progenitor plant cell. And then this diverged in three different ways. It formed three lineages: one, the glaucophytes; the other, the green algae, from which later the vascular plants arose; and then the red algae. About 1 billion years ago, there was a secondary endosymbiotic event where a different heterotroph engulfed the red algal cell. And over time, the nucleus and the mitochondria from the red algal cell were lost, and the chloroplast was retained, and we have now the structure of what is typical for a diatom. Now, diatom chloroplasts are different than those in green algae because they have two additional membranes surrounding the chloroplast. The periplastid membrane, which is a remnant of the red algal plasma membrane, and then the endoplasmic reticulum, which is a remnant of the engulfing event. And so they have some distinct features at that level. But an important point here is that there's 1 billion years of distinct evolutionary history comparing a diatom with a green algae. So this is a very complicated slide, but there's a summary of some of the points here. We just went through and we've highlighted a number of differences between the green algae and diatoms. And I'll just summarize some of the points. The process of photosynthesis is very different. The way that they process carbon in the cell, the location in the cell where they make nucleotides and other biochemicals is different. Bottom line is not only do they have different appearances, but they're very, very different metabolically. And we think this might have something to do with the differences in productivity in different classes of algae. So diatoms are extremely productive on a global scale, and one of the questions we can ask is, could these metabolic differences account for their high productivity? So diatoms are so abundant you can actually observe them from space. On the left, we have a satellite image of the Southern Ocean, and we're monitoring chlorophyll levels by satellite, and diatoms are especially abundant in the Southern Ocean. On the right is an image of Vancouver Island in Washington State, and you can see off the coast there's a bloom of diatoms. So this is a vast scale. And diatoms are responsible for capturing about 20% of the carbon on the planet as CO2, generating an equivalent amount of oxygen. And this is actually more than the world's rainforests. So I'm going to focus on a couple areas that are different between the green algae and diatoms that might relate to differences in productivity. So in photosynthetic eukaryotes, carbon is fixed as CO2 from the atmosphere by an enzyme called rubisco, and it forms a compound called 3-phosphoglycerate. And through a series of steps, the 3-PG is converted into carbohydrate through the process of gluconeogenesis. When the carbohydrate is needed by the cell, it breaks it down by the process of glycolysis, and it can generate energy and biomolecules that the cell can use. So in the chlorophytes, they store their carbohydrates in the chloroplast. So they will take the 3-PG, perform gluconeogenesis, and they form insoluble starch, which surrounds a structure called the pyrenoid in the chloroplast. In diatoms, they export the carbon from the chloroplast into a special compartment called the chrysolaminarin vacuole. And rather than storing starch, they link the sugar molecules together differently in what's called a beta-1,3 linked glucan called chrysolaminarin. And just to give you an idea of the size in which they can store this material, on the far right is a cutaway view of a diatom cell showing the size of the chrysolaminarin vacuole relative to the chloroplast. Now, there are differences not only in the location where they're storing the carbohydrate, but also the accessibility is different. Chrysolaminarin is water soluble, it's easy to break it down. Starch is insoluble, takes more energy to break it down. So you can ask the question, do the differences in the form of carbohydrate that's stored, their location, and their solubility affect the ability of the cell to use this carbon? Related to that is the pathways of carbon flux in the cell. So on the left we have a diagram of a model chlorophyte, Chlamydomonas reinhardtii. And chlammy, as it's called, stores its starch and breaks it down in the chloroplast. And if it wants to use that starch as a starting point for energy, it breaks it down in the chloroplast, exports it through the cytoplasm into the mitochondria to generate energy. In a diatom, because they store their carbohydrate outside the chloroplast, they have a couple of different options in terms of how they can process that carbon. They can choose to store the carbon as chrysolaminarin if they want to, or they can use it directly and import it out of the chloroplast into the mitochondria and use that for division. Now that gives them some flexibility. One of the issues that might come up if you're doing both glycolysis and gluconeogenesis in the same compartment is they're competing reactions. They're going in both opposite directions, basically. And so to accomplish that, you might need to have more control over the process or have conditions where you're favoring one process over the other at different times. And diatoms seem to be able to avoid that. And you look at this, in fact, diatoms actually have glycolytic or gluconeogenic pathways in 3 different compartments in the cell. And you might think, well, this is very complicated. It must not be very efficient. But it's important to realize that simplicity isn't necessarily correlating with efficiency. And in fact, a more complicated process could be more efficient. And I use the example here of running shoes. So on the left, we have a relatively simple running shoe. It's perfectly functional, works well. But compare that with a highly engineered running shoe where the forces that are involved are modeled and optimized to make you run faster. And so that might be what is going on in this comparison. This idea of storing carbon in different places and it's somehow affecting the productivity of algae seems to be established in evolutionary sense. So this is a diagram of different classes of algae over evolutionary history, and we'll start with the cyanobacteria on the upper left. You have progenitor chloroplast, the initial primary endosymbiotic event forming the glaucophytes. They store their carbohydrate as starch outside of the chloroplast. The green algae, the chlorophytes, imported all of the machinery that makes starch from the cytoplasm into the chloroplast. So that's a major metabolic shift, and there must be a good reason for doing that. The red algae retain the formation of starch or glycogen even in the cytoplasm. They use a slightly different form of starch. When we look at the secondary endosymbionts in the lower panel, The euclinophytes, a green algal secondary endosymbiont, stores a water-soluble beta-1,3-glucan in the chloroplast. So basically what they've done is start where the chlorophytes are, where they move the starch synthesis into the chloroplast, then moved it back out. So obviously there's a good reason for that in terms of productivity. The chloracneophytes, another green algal symbiont, have a very unique structure. They store, again, a beta-1,3-glucan in a structure outside of the chloroplast surrounding something called the pyrenoid. When we look at the red algal symbionts, secondary symbionts, the cryptophytes store starch. So they decided to use starch in this case in a somewhat similar location. And then we talked about the diatoms and chrysolaminarin. So over evolutionary history, these different classes experimented, if you will, with storing the carbon in different forms and in different locations. And we think this affects their overall productivity. The diatoms are interesting for another reason. They're cell walls. So this is an image of a single diatom cell not treated with anything. But if we treat that cell with acid, which degrades all of the organic material, what's left is a hard shell of silica. And these organisms have evolved the means to take up dissolved silicon from the seawater, transport it into the cell, and they can actually harden it and make a mineral of silica. And silica is the exact chemical composition of glass. So these organisms basically make a cell wall out of glass. And this process is called biomineralization. They've taken a inert material in the ocean and made a mineral structure out of it through biological processes. And diatoms are extremely good at doing this. These are different images of different species of diatoms, and they've been called nature's nanotechnologists for their ability to form a whole variety of these structures on very small scales. And this is a subject of a lot of focus research studying how they make these structures. One potential factor involved in this is because the cell wall is made up largely of silica, diatoms need less carbon for their cell wall. So potentially that also contributes to their higher productivity. They can use the carbon that's normally used for other purposes, making the cell wall, and repurpose it for growth and division. Thank you.